Good day, this is Professor Will, CBMD PhD. Today is Tuesday, June 15, 2021. It is 8:03 and 37 seconds a.m. Eastern Standard Time. Today's research, pancreatic tumors. And don't forget to check out my website. So let's hop to it here. Targeted drug found effective in thwarting pancreatic tumors. Date June 14, 2021. Source, Virginia Commonwealth University. Through a preclinical study, researchers identified a novel drug that effectively thwarts pancreatic tumors that are addicted to the cancer-causing mutant KRAS gene. Pancreatic cancer is an aggressive disease in which malignant cells form in the tissues of the pancreas, a long and flat gland located behind the stomach that helps with digestion and blood sugar regulation. Because pancreatic cancer is difficult to detect early, it is associated with low survival rate, according for just over 3% of all new cancer cases in the U.S., but leading to nearly 8% of all cancer deaths, according to the National Cancer Institute. Researchers discovered a link between hyperactivation of the CDK protein and mutant KRAS addiction. Researchers exploited this link preclinically to counter mutant KRAS-driven pancreatic cancer, warranting clinical investigation in patients afflicted with this deadly disease. Research findings are highly significant as they revealed a new avenue to combat an aggressive form of pancreatic cancer with very poor prognosis, due mainly to its resistance to conventional therapies. KRAS is mutated in 90% of pancreatic cancers. Previous research from the lab has demonstrated that some tumors that harbor mutant KRAS are actually addicted to the mutant gene, meaning they can't survive or grow without it. Researchers set out to discover if there is a drug that can specifically kill tumors that are addicted to mutant KRAS. First, they mapped out the blueprint of pancreatic cancer cells through global phosphoproteomics, which gave them a snapshot of how the addicted and non-addicted tumors differ at the phosphoprotein level. Researchers found two proteins, CDK1 and CDK2, which were indicative of which cells were addictive to mutant KRAS. Additionally, they analyzed a comprehensive database from the Broad Institute of MIT and Harvard that contains genome-wide CRISPR gRNA screening datasets. They found that CDK1 and CDK2, as well as CDK7 and CDK9, proteins were associated with mutant KRAS-addicted tumors. Lastly, they evaluated the ability of a library of 294 FDA drugs to selectively kill mutant KRAS-addicted cancer cells over non-KRAS-addicted cancer cells in the lab and determined the most effective drug in preclinical experiments was AT7519, an inhibitor of CDK1, CDK2, CDK7, and CDK9. Using three entirely different approaches, the same conclusion presented itself clearly to us. Pancreatic cancer patients whose tumors are addicted to mutant KRAS can benefit greatly from treatment with the CDK inhibitor AT7519. To further validate these findings in fresh patient-derived tumors from pancreatic cancer patients, they found that AT7519 suppressed the growth of xenograft cells from 5 mutant KRAS pancreatic cancer patients who relapsed on chemotherapy and/or radiation therapies. AT719 has previously been tested unsuccessfully in a number of clinical trials, but none of the trials targeted pancreatic cancer. If the research findings are correct and translate in humans, then researchers should be able to see a positive response in pancreatic cancer patients whose tumors are addicted to mutant KRAS. In addition to pancreatic cancer, these findings may also have clinical implications for colorectal and non-small cell lung cancer. Patients where mutations in KRAS are prevalent. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.